Good morning, everyone. Welcome to Campus Friday. Today, our speaker will be Chelsea Lopez. Chelsea is a fifth-year PhD candidate in Ocean Sciences Department, studying dissolved organic carbon dynamic. She currently lives in Washington, D.C., and she is working at NASA's Ocean Ecology Lab. Chelsea is also awarded with 2021 Best Student Publication Prize, also known as Miller Prize, in recognition for paper Evidence of Deep Dock Enrichment by Particle Export, Subarctic, and Northern Subtropical Front in the North Pacific. Today, Chelsea will give us a talk about the influence of gelatinous zooplankton and turbulent fronts in deep carbon export. Chelsea, please take the stage.
Thank you, Payson. Sure. Okay. You can still see the laser pointer, right?
Yes.
Okay, great, thanks. So yeah, I'll be talking about gelatinous zooplankton and turbulent fronts and how they impact deep carbon export and then ultimately can take DOC to the deep ocean. So just a quick outline so that you know what I'll be talking about and you can follow along because I have two parts here that are a little different from each other. So first, I'll be talking about those fronts and how they're exporting carbon to the deep ocean and evidence of that, the particles and DOC. And then I'll be talking about the mechanisms behind that, so the biogeochemical controls on that export and showing some evidence of why mixo depth and physical factors play into that. And then part two, I'll talk about seasonal and interannual TOC variability. And I kind of tricked you with my title by putting gelatinous zooplankton so that you would show up because that's pretty cool. And then how the export from them release DOC at depth. So I want to frame this a bit about why we care about this research anyway, because I feel like when I listen to talks and hear people talk about the research, oftentimes they narrow it to one specific field or one specific topic, and we don't really talk about the big picture sometimes. And I think that's always kind of bothered me. So I think that this research is important because we want that baseline understanding of the carbon cycle and the stocks and what's going on the Earth. And that's going to be important as the climate changes. So I like to frame my research in that way. So I like to focus on the deep ocean because that carbon is sequestered for longer periods of time. And so those are important processes for long-term carbon storage, and climate change will impact that. So just a quick overview of the biological pump and how carbon makes it to depth. I know most of you probably know this already, but for any of you that don't. So CO2 goes into the ocean from what we've emitted, and it's there in the surface layer. And then the phytoplankton take that up and produce oxygen, and they are constrained to the upper 100 meters roughly in the mixed layer, which is the top layer in which the water is stratified, and it's that top layer that water can mix freely above that. And so zooplankton can they consume the phytoplankton, and they are transferring carbon from that phytoplankton into larger forms of life that transfer carbon into bigger particles. So you can think of a zooplankton itself as a particle. And that transfers into higher trophic levels. And so when you have nutrient-rich areas, oftentimes you have larger phytoplankton, you have more of them, they transfer carbon into larger and larger trophic levels, and that's important. And so those carcasses in their fecal matter are heavy and they sink. And so when you have higher trophic levels and a lot of carbon, you're going to get more particles, and they're heavier, they can reach the deep ocean quickly. And so what I'll talk about here is how those sinking particles have the capability of releasing DOC, dissolved inorganic carbon, and in the very deep ocean, the bathypelagic depths. And so that happens by the particles dissolving, microbes attach to the particles and release DOC in that way. And then that dissolved organic carbon can either be added to the long-term, the refractory dissolved organic pool and be sequestered for long periods of time, or microbes can consume it and turn it into dissolved inorganic carbon, which contributes to that biological pump. So now you're putting carbon in that form that's going to last for longer periods of time. So it all comes back to deep carbon sequestration. So here are the sections for the two parts. I'm focusing in the North Pacific. So for part one, I'll be talking about this meridional section here, and a transect that we have bottle data from and some sensor data that'll show you to explain some of the exports that's going on because of fronts that pass through here. And then in part two, I'll talk about seasonal and interannual changes in carbon along here in the Gulf of Alaska. So two different topics going on, but in the same area. So here we go, part one, fronts enriched. We have evidence of it, deep DOC through sinking particles that make it to bathypelagic depths. So first of all, what are fronts? Turbulent water areas, they can be small or large. They can traverse several degrees in latitude, or they can be small and last a few days. And they differ in the depth in which they mix water. So here's what one might look like. And so these are a result of abrupt changes in salinity, temperature, density, or a combination. It's usually a combination because they're all related. But you can't always have a salinity front matching with a temperature front. And especially in the North Pacific, that's important because there's salinity temperature compensation, which I won't get too much into, but for the North Pacific, you have to use salinity as a tracer for these larger fronts that I'll be talking about. So why care about fronts at all? They have the capability of mixing that high nutrient water from depth into low nutrient water at the surface. So it's replenishing the surface with nutrients, and that supports phytoplankton. So if they get nutrients, they're happy, they grow, you get larger phytoplankton, you get more of them, and you're transferring carbon into higher trophic levels. And then that produces larger particles, and those larger particles reach depth quickly. So good for phytoplankton growth. So part one, again, looking at this transect here, it's called P16N from the GO SHIP program. It's a long-term hydrographic program. They collect CTD data, they collect bottle data, and this has been going on for decades. And then I want to point out that the one transect that I'm focusing on here, because it'll be important, is that this transect is from springtime. So I'm also looking at UVP data, which is the ultravision profiler. And this is attached to the CTD, which I'll show you a picture of to show you what field work would look like for this. But it's a sensor that they attach to the rosette. It goes down, it takes a lot of images of particles, and then they can analyze that and turn it into different parameters. But I'm looking at particles per liter, depending on size classes. They can also look at biomass and different zooplankton classes, things like that. But there's some labs that they use this data, and it's publicly available on Ecotaxa if you ever want to look at it. And then I look at some satellite chlorophyll data as well to kind of confirm some of our suspicions and our explanations for the biogeochemical controls. And then lots of thinking. So here is what a CTD looks like. You collect water from the bottles. They go down, they trip at different depths, so you can collect water from lots of different depths. So to collect DOC, we place an inline filter off the spigot of a rosette bottle, of the Niskin bottle, and then put it into these vials, which we then ship back to the lab and measure on TOC analyzers. And then this cage that's attached to it is the UVP. So that's the sensor that's taking those images. Here's what one of those images would look like. That's actually a jellyfish. So it's measuring life that's down there, particles, measuring that, converting it into particles per liter, that data, that's important. Okay, so how to locate these fronts using the profiles from the CTD, so your salinity profiles, things like that. So here I'm showing you the transect, and I'll go through the sections in a minute, but I'm just trying to show you how you would identify one. So these are the contours. So this would be like a salinity, one salinity. And so you have this isohaline, they call it. And so you can see in certain areas where these collect. So if you have abrupt changes in salinity, it would look like this. And you can see there's quite a few here in this section. And then some of them reach deeper depths. Some of them, you know, cut off here. So they're all a little different. So this is how you would identify it. And then this area is also very well characterized physically because there was a researcher from the 1970s who focused on this area specifically, pointing out what numbers, what isohalines are important. And they match up really well, even though it's been 50 years. Okay, so I'm gonna point out how the salinity profile matches up with the locations in this transect. So first you have the subarctic gyre, right? And then that is over here on this profile. And then your subtropical gyre is over here. And I apologize for all the animations, but I want to make sure that you can understand the sections well and see where I'm going because there's a lot going on. So there's this section here that is called the subarctic frontal zone. And so this is not a front itself. That is important. It is an area where fronts often pass through. So it's an active area. So there might be eddies coming through here. There might be short-term fronts. It's turbulent water. And then fronts that I will be talking about pass through here. So that is in this section here. And so that's characterized. So you have this cut off here, and then I had to use some past data to figure out this edge of it. So this is the whole zone. And so you'll have eddies coming through here. You can see this is a front. And then you have the subtropical frontal zone here, which is right here. So you'll have subtropical fronts. So these are the fronts that pass through this area, these areas. And then you have the northern subtropical front here, which is right on the edge of this frontal zone. And then you have the subarctic front, and that's around 41, 42 degrees latitude. And then these ones can move around. So they don't, climatologically don't move more than 1 or 2 degrees in latitude. And they're pretty consistent in that. So here's what those fronts would look like passing through. So I'm going to focus on these three because they are the largest ones. They are the ones that seem to be impacting the biogeochemistry of these areas. So to make this easier for you to look at, I'm going to flip this. Okay, so this lines up with the profile. You've got your 25 degrees north is on this side, 50 degrees north is on this side, and then your sections pass through here. So first is the dissolved organic DOC profile. And so you can see there's 2 columns of enhanced DOC concentrations that are vertical. So they must be coming from vertical export. And then here's small particle data. And when I say small particles, it's subjective. So this is within the limits of certainty of the UVP. So you would need a different sensor to measure particles smaller than that. Like they call them like lists, different benchtop things that they take to see those sensors. So it's subjective. So 0.1 to 0.5 millimeters, which is kind of relatively large compared to those micrometer particles. And then here's large particles, so greater than 2 millimeters, so relatively large. And you can see between all 3 of these profiles, they are different. So I want to figure out why. So I'm going to point out the observations, and then I will try to explain to you why I think that you see these profiles. So here, this is a subtropical front. So right here in the transect, you can see that there's not a lot of DOC enrichment. It's not like one of these columns. And then from what we can see, there's some missing data here. No enrichment in small particles, no enrichment in large particles. So if you move to the northern subtropical front, you can see there's an enrichment in DOC, a very significant one. And then small particles, there's some here, but it's not making it down to depth from what you can see. And then the subarctic front has the enrichment in DOC here as well. You have an enrichment in small particles, and you have an enrichment in large particles. And then just to point out DOC concentration, so these dots are every single observation. And then you have, this is your mean, and 50% of the data falls in here. So just showing between the sections, here's your Northern Subtropical Front, here's your Subarctic Front. Your concentrations average are much higher than the regions around. So here I'm going to try to explain what the biogeochemical controls are on this area. So here is the nitrate profile in the upper 150 meters. And so nitrate is important for supporting the phytoplankton. So they need that nitrate to survive, they need it to grow. It's involved in spring blooms. So here's the one micromolar contour. So this would be the first area where phytoplankton can reach enough nitrate to grow. So that's a neutrocline. And then here I'm just showing you that DOC profile again, and this is deep ocean. I'm focused mostly below 1,000 meters. So this would be your bathypelagic water column. And then remember, you have these two vertical enhancements in DOC. So, and then I'm pointing out here each of the three fronts: turbulent water. And then here's the winter mixed layer depth. So remember that these observations are from springtime. So I'm going back in time here. And right here is where the winter mixed layer depth crosses the neutrocline. And this is important because this is in wintertime where you're, remember that this is water that's mixing regularly. So you have nutrients here that are able to mix through here. So that means phytoplankton are being supported in winter. So that would logically induce carbon export, and you would have export coming down here, and then that would be releasing DOC. But remember that these observations from springtime did not show particles in this area. So that would suggest that if you have export going here in winter because you have enough nutrients, by springtime, you can't see those particles anymore, but you can see the remnants of the DOC that it released into the water column. So it gives you a hint of like what the residence time of those particles is from one season. So they don't make it through the seasonal from winter to springtime, but the DOC does. And so as seasons go on, you have your winter mixed layer shoaling, and then now you have your spring mixed layer depth. So now you're farther away from access to those nutrients. So in spring, you now have the mixed layer depth crossing the neutrocline here around 42 degrees north. And so this is where we also see that enrichment in DOC. And we did see large particles and small particles. So that would suggest that that export is happening instantaneously. Maybe it's also happening in winter. It would make sense that it is. We don't have evidence of that. So just to kind of summarize each of these, here in the subtropical front, even though it's front, it's not showing nutrients. Oh, sorry, it's not showing particles of either size class or a DOC enrichment. But if you notice here, the winter mixed layer depth doesn't even reach the neutrocline. So at no point are you getting enough nutrients in here to support phytoplankton and that transfer carbon into higher trophic levels that would induce carbon export and release DOC. So that makes sense. And then your Northern Subtropical Front, you have enough particles, you have enough nitrate nutrients to support life here during winter, export those particles that release DOC, but you don't see the particles in springtime because the winter mixed layer shoaled. And then here you have both wintertime crossing the neutrocline, so enough nutrients in winter, enough nutrients in spring. You're getting all those particles coming down, and then we have evidence of it releasing DOC in that way. So here, even in winter, it crosses the neutrocline and summer. Oh, sorry, not in summer, but in winter. So you would suspect that you would see export in this area, but this area is not within a frontal zone. Oh, sorry, it is within a frontal zone, but does not have a front associated, not one of these larger fronts. So we see those small particles coming down, but we don't see the large particles. We don't see DOC. So that would suggest that these larger particles are more important for releasing DOC into the deep ocean. So even though this makes sense logically, we want to find evidence that shows that this hypothesis is real. So here I'm going to define what I call the chlorophyll boundary. In the literature, it's called the transition zone chlorophyll front. But I wanted to get away from calling it a front because it's not a physical front. So I don't want to confuse anyone. So it's chlorophyll boundary. So if we track this boundary using satellite data, satellite chlorophyll data, we can track the seasonal movement of eutrophic conditions. So it would be like where that mixed layer depth crosses the nutricline, we can track that. It's not a permanent location, so it changes from winter to spring. So we can see if that matches up with what we think, those locations that we think it's crossing. So it's operationally defined as a 0.2 milligram per cubic meter isopleth, so just where that threshold is crossed. And so here, 150 degrees west, this is where our transect is. So it's kind of hard to tell. I'll point it out in another image, but it's somewhere around here. And that lines up where the Northern Subtropical Front is. So that makes sense with our hypothesis. And then here in spring, you have it moving back. You know, your seasonal shoaling of the mixed layer pushes that boundary back. So here you can see it crossing around 40, a little bit north of 40, which is exactly where we thought it would be. So just to make that a little more obvious, here's where it is in winter, right on the Northern Subtropical Front. It moves north, right on the Subarctic Front here. So it lines up perfectly, great. And then some other evidence that is interesting is through this protein called rubisco. And so it is quantifiable, so you can actually measure this, similar to as you would measure DOC, but it traces modern carbon to the deep ocean. So this is a product of biological processes. So it'd be like a modern process that happened recently. And it is within microgels, which is kind of like a dissolved gel complex particle type thing. It's not particles, but it is, this is microgel. So there's a profile here from a paper by Monica Oriana and Dennis in 2012. And so they can see an enrichment of that. So that would be evidence of recent export. And that's right where the subarctic front is. So that's really interesting. You can see that even in past data. And then this is the same transect that I'm looking at here. It's just from 2006. So conclusions from this data and this analysis, we saw that deep export of particles was occurring, and then we saw DOC being released in those enrichments. And it seemed to be dependent on the presence of a strong physical front, but we do not see it in one of the fronts. And why is that? It appears that you need the proper biogeochemical conditions, so the mixed layer depth that crosses the nutricline, so you need to have the access of nutrients in the mixed layer to induce export. But if you have that front there, you are basically injecting nutrients longer and more consistently into the mixed layer. So you are increasing the rate of export, which ultimately appears to release DOC. So going forward, why do these observations matter? Why does this analysis matter? What can it do for the scientific community? So it just highlights a large-scale process that hasn't really been discussed in terms of deep export, which is important for carbon sequestration. And then this analysis just kind of leads to more questions. What's going on? So what are the temporal characteristics? We don't really have the data to explore that right now. So how would it change between years? How does it change in 10 years? There are big climatological events that happen through that area, like the blobs, so heat blobs that go through the Northern Pacific. How would those affect it? And should models include frontal export? Because here we're looking at one transect, but these fronts cross the entire basin. So that would be a lot of export going on. So do we need to include that in models, and how important is it for our carbon budgets? And then will it change as climate change? So there's some previous work looking at these sort of processes. A lot of it is in the mesopelagic, so 500 meters and above. And there's some work in the California current going on. There's some work in the Kuroshio current, but there's right now no studies looking at the evidence of deep export. And there's no studies looking at how that affects the DOC at those depths. So I'm happy that we can highlight that that is going on. All right, so here's part 2. And this is where I'll talk about the gelatinous zooplankton. I know you're waiting for it. So this is seasonal interannual TOC variability. So in one section, how is it changing seasonally? Does it matter? How does it change between years? And then some zooplankton that I'll show you. So here's the study site, northeast of where I was showing you previously. And so this is a Line P program, which is operated by the Department of Fisheries and Ocean from Canada. So they use Canadian Coast Guard vessels to do this 3 times a year. So winter, spring, summer. They do your basic oxygen, temperature, salinity measurements, and they do the rosette bottle data. But there hasn't really been any organic carbon measurements going on in this hydrographic program. So the Hansel lab collected samples in 2017 and 2018 in all 3 seasons. And I want to bring your attention to an issue that I think is important. So you might have noticed now I'm calling it TOC, which is total organic carbon. So whole water samples are taken at great depths typically, but they are often considered equivalent to DOC. So it would be greater than 90% DOC in those vials, but they still may include particles. So when you filter it, then you're considering it true DOC. So anything less than 0.7 microns. But in this case, I'm going to show you why it's kind of dangerous to assume that TOC is always equivalent to DOC. So some questions I have here. How do those concentrations change seasonally and interannual, obviously? But what is stimulating that export, those changes that we see? What's the reason behind it? And does that export release DOC in its wake, which is really what I'm interested in. So we'll go through the observations with you real quick. So 2017, I'm looking at fluorescence data. And so that's sensor data, but you can assume it's correlated with phytoplankton concentrations. So here's wintertime, 2017. I'm looking at the upper 150 meters. And there's some modest concentrations going on there. And then springtime, you can see the concentrations increase. There's a development of the chlorophyll, deep chlorophyll maximum going on here, which is consistent with what a spring bloom would look like. And then in summertime, you can see there's a lot more chlorophyll here. There's some sort of bloom going on. And then here's the profiles of total organic carbon. And then this is in the bathypelagic. I'm really focused on 1,000 meters and below. So you can kind of ignore this, but I wanted to give a better picture of what it looks like. So you can see winter's pretty calm, and then spring comes along and you have more heterogeneity. So you have these hot spots going on, which makes sense. There'd be export happening from springtime, making it down to the deep ocean. And then by summer, you can see more of the heterogeneity. So deep export going on, releasing DOC, particularly here where you have high levels of phytoplankton. So we can assume that the increased fluorescent is correlated with the increased TOC hotspots through carbon export. So here in 2018, this is pretty similar to 2017, a little bit higher concentrations on this side though. And then spring, there's a lot going on. These concentrations are very, very high. And then summer, it starts to taper off again, just as we saw in 2017. And you can see there's a lot more heterogeneity here. You have more hotspots, you have a lot going on. So higher export occurring, releasing DOC. And then here, these are really, really high values. Dennis took one look at it and said that's contaminated. And that was a little concerning. So maybe you need to throw that data out. I don't know. But I will talk about that in a second. And then here you can see it tapers off again. But these enhancements are pretty similar to what's going on here. So it seems that whatever export happened here is being consumed by summer. So you might just be listening to me say, oh, this is variable, but I wanted to show you some data that would kind of put that into context. So here is Aloha Station that's near Hawaii. And so you can see between the 3 seasons, it's pretty consistent, those concentrations between each other. You see it in spring here, there's a little bit of export going on there. And then the red line is just a baseline for you to see, okay, this is lower concentrations, we've got higher concentrations over here. But you can see how variable this is compared to over here. So a lot of variability. All right, back to this big bloom in 2018. I wanted to see if I could figure out why this is happening, if there was evidence of any additional process that we're missing here. So first I looked at satellite chlorophyll, and you can see here's 2017, and this is the transect here, and compared it to 2018, and you can see chlorophyll levels are much higher. So that's encouraging. And so the Line P program does, they do net tows here. So these are called bongo nets. They send them down to 250 meters, bring them back up, it's catching those creatures. There's a filter here. So what they'll do is bring it up and then put anything that's caught into a bucket, and then they start sorting through them and counting with whatever's in there. And one of the things they count is gelatinous zooplankton. So here's a salp, and it kind of scoots through the water. And as it does that, it's collecting particles within itself, and it consumes those particles. So that's what it's eating. And these can get pretty big, like the size of your hand, and they also form chains, really long chains. I've seen people eat one. I don't recommend it. So here is some data from the 2 years of P26, which is the most offshore station. In 2017, there was none. 2018, you see higher counts of them. If you think about it, so this is number per cubic meter. If you have 4 of these per cubic meter, that's a lot. So I know these numbers don't mean anything to you really, but you can believe me, say it's a lot. And then here's larvaceans. I think these are my favorite. So this is the actual creature here. You can see its tail. And so it builds this house around itself. So this net around itself isn't actually part of it. And so these are a couple millimeters in size. And so they also collect particles within itself. But as they do that, it can get clogged wherever the intake is. I'm not quite sure on this photo. So when those particles are coming in, this they call it the house, the gelatinous house, can get clogged, and these creatures eject out. And so when they do that, they leave this house to basically sink out. So they're contributing to export in a unique way that way by exporting those houses out. And so here is the same. There's a lot of them. There's a lot of them here in 2018 versus 2017. And if you look at the climatological data from years before 2017, the numbers look like this. This is a highly anomalous event that happened in that season. And then doliolids are out there as well. They're pretty similar to salps, but much smaller. So also only a few millimeters in size. And the same thing, there's a lot of them in 2018. And they are dominating. So if you have 81 per cubic meter, that's a lot. So, but does export from these creatures, does it release DOC, and can we show evidence of that outside of the fact that there's counts of them? So here's the TOC profile that I showed you previously. And so here's those really large numbers. Maybe you have a whole lot of those creatures around here. Here's also what looks like an export event. And so in the season, we happen to collect both whole water, so your TOC samples from the Niskin bottles, and then filtered water. So it's filtered at every single depth, versus these would be unfiltered and assumed to be DOC concentrations. But you can see the difference between the two. So here's 119 micromolar, here's 54. So that's showing there's a good amount of particle in that specific sample. But the fact that this DOC sample is also elevated suggests that whatever particles are coming through here are releasing DOC. And then this station happened to be the one where the majority of the salps and the larvaceans were located. So main point, it appears that whatever's released, the export from higher phytoplankton levels, higher gelatinous zooplankton levels, is releasing DOC even in the great water column, even at these great depths. And it seems to be within relatively quick time scales. So we're talking like days to weeks. So just to show and kind of move outside of that topic, the zooplankton, about accumulation of carbon, so accumulation of that TOC. So I averaged the profiles of the section that I was looking at, and I'm showing the two seasons here. So in winter, they're pretty within the realm of uncertainty, pretty much the same between 2017, 2018. We look at springtime, which makes sense from our observations. You have that enhanced variability of both, really. And springtime 2018, the concentrations were higher. They enhanced higher. So you've got quite an offset here. But the patterns are the same, which is interesting. And then by summer, it all comes back down. They're starting to converge again. So that leads me to accumulation rates. So here's the rate of accumulation and removal on order of days. So how much is being accumulated or removed each day. So here in 2017, that carbon accumulated at 0.5-ish micromoles per day. And in 2017, the removal rate was pretty similar. And then you could see the same thing with 2018 between accumulation, removal. So even though there was a lot more accumulation, they both had nearly equivalent rates of accumulation and removal. So this would suggest that that modern carbon that's being exported to depth is being removed nearly entirely by the time summer rolls around. But how is it removed? You can have those particles sedimenting out. You can have respiration by microbes into the dissolved inorganic pool, and ultimately sequestering carbon in the deep ocean. So some conclusions from that data. We wanted to know if TOC concentrations change seasonally and interannually. And the answer was yes. So we saw it change between winter to summer. We saw it accumulate. And we saw it accumulate from winter to spring and then be removed from spring to winter. We saw a difference between that process in 2017 and 2018 pretty significantly. So you had a big bloom in 2018 that you didn't observe in 2017, which appeared to be connected to the gelatinous zooplankton and higher levels of phytoplankton as well. So what was stimulating that export? Enhanced levels of phytoplankton, enhanced levels of zooplankton, which were capable of exporting DOC or exporting particles that release DOC even at great depths. And then we wanted to know if that export released DOC, and we saw evidence of that through the difference between the TOC and the DOC profiles. So I want to thank you guys for showing up today, and I want to thank my advisor, Dennis, and Lillian and Jennifer for all of their help over the last few years. And then if you have any questions, I'd love to talk about it.
Thank you so much, Chelsea, for doing this wonderful presentation. For our audience, if you have any questions, please turn on your video camera and unmute yourselves and ask our speakers if you have any questions.
I'll go ahead and start, Chelsea.
Surprise.
Yeah, someone's got to break the ice, right? I know John will have a question eventually. So great presentation. You know, you really did a nice job of explaining the frontal system, and it was a pretty complicated system. So it was nice that you took the time to do that. So given, you know, all the uncertainties, you know, all the questions you could have about that system, and you mentioned, you know, a few of them, like, does it, you know, follow the front across the basin, you know, what are the actual mechanics of those fronts, you know, and, you know, their role in export and all that sort of thing. Is there a priority question for you when you think about, boy, if I could only get a ship to go out there and do XYZ to answer these questions, and you can't do it at all, right? So what are you most interested in or curious about, or what would you want to pursue?
I would think there's like two answers to this. My first would be because those currents cross the entire basin. I think the thought of, oh, is that export happening on that scale across the whole basin? Is it stronger? I think I read something that said those fronts were stronger on the west side. So that could mean that there's higher export over there. I think that would be really interesting to know. And I think that comes back into like, should we include that in models? And how does it change with, I know that was close to a blob year with a blob event. So does that matter? It's in that area. I don't know. I guess I have a lot of questions about it, but I think the fact that it crosses the basin would probably be my biggest interest, because it'd be easier to get ships to go there all at the same time and cross that. Maybe.
Okay. Go ahead, John.
Yeah, thanks for a great presentation. I thoroughly enjoyed that. And the importance of jellyfish is greatly underestimated. With respect to frontal surfaces, they are often sloped and horizontal. They're not a vertical feature so much. And so I wonder, I know you're making your deductions from bottled data and so forth, which is basically collected in a vertical profile. And but it might be neat to look at some of the factors that essentially follow the frontal surface in a tilted way. I don't know exactly how you would go about doing something like that unless you had something like an autonomous vehicle that could follow the frontal surface. The other thing that would be neat is to see using towed bodies to make more vertical profiles as you're going along. Now, of course, you're limited in depth as to how far you can go down with those things while you're underway, but that might be kind of another way to slice the salami.
Yeah, that would be super interesting. I didn't think about that. I so it would depend too a bit on the scale of the slope. Like, is it a couple kilometers? Is it not that sloped? I think it would really depend.
Yeah, probably tens or hundreds of kilometers in terms of the region where the slope is occurring. You know, if you follow it down to depth, and of course a lot of things move along frontal surfaces. They're isopycnal surfaces oftentimes as well. So natural spreading tends to follow isopycnals, and so that's another reason for following the frontal surface itself. Anyway, well, fantastic talk. I thoroughly enjoyed it.
Thank you.
I'm amazed that you were able to get so far with limited amounts of data.
What do you think of those? There's a lot of data, John.
Yeah, well, a lot of data to process, but compared to the scope of the ocean,
yeah,
very limited.
Any more questions from our audience?
Well, I'll ask one. So what's next? What are you working on?
As far as the dissertation or?
Your science.
Science. Okay, I'll talk about dissertation then.
Yeah.
So I am looking at a project that goes through the countercurrent system in the Southern Ocean. So similarly looking at how maybe physical perturbations are affecting export in that area. So there was a profile that Dennis and I were looking at between, I think it was 2007 and 2018. So within a decade. And so you would see a lot of DOC exported to the deep ocean in one year and not the other. So then it just made me wonder what physical factors might be involved in that. And it's right in the belt of those currents. So I think that's interesting. Was there an event that happened there? Was it just a strong storm season? I don't know. So that's what I'm working on.
Very nice presentation, Chelsea. Thank you.
Thank you, Dennis.
Well, thanks again, Chelsea. That was great.
Any more questions for our speaker? All right, so if there's no questions, thank you so much again for attending Chelsea's presentation. Great work, Chelsea, by the way. Our video will be recorded and will be posted on YouTube. So if you are interested in some details, feel free to check those video recording on our channel. And thank you so much for attending. Happy weekend to you all.
Thank you all.
Bye-bye. Thank
Bye-bye.
you.